I think there's many reasons to apply analytical quality by design. I think primarily the main reason is to ensure that we have robust analytical controls which measure critical quality attributes to the required uncertainty, so we can sentence batches with confidence, which ensures patient safety and efficacy. So I think that's the primary reason. It really drives robustness. I think secondary reasons is very much from a manufacturer perspective. I think it enables us to get an understanding about our method performance, and it enables us to maybe deliver a framework to understand alternative ways of measuring CQAs, so we can maybe switch between different analytical procedures and different analytical technologies in a more agile way. So I'm hoping this new framework is going to enable greater innovation.
We've taken a very practical case study. So we took a cross-agency group. So this involved license assessors from the licensing division of MHRA, inspectors, GMP inspectors from MHRA, folks from our laboratories, and folks from the Pharmacopoeia. And we took a product, atorvastatin tablets, and we tried to apply some of these concepts. Think about how it related to methods in the Pharmacopoeia.
Expectation is that these approaches can support new methods, new products, but also can be incorporated into existing monograph when we are able to define the basic element.
So I think we should be judicious with how we apply the tools, and I certainly wouldn't recommend spending a lot of effort on very simple, maybe well-established pharmacopoeial methods where there's a very large amount of space between where the product is running and specification limits. If there's no risk to the patient, if there's no real need to fix something, then I wouldn't recommend doing anything. We really should use a risk-based logic to decide what we apply these tools to. And the whole idea of post-approval change management protocols, or PACMPs, is to really agree that up front between industry and regulators, so we are more predictive with changes. And the hope from an industry perspective is we lower that post-approval change burden by providing science and really well thought out protocols for what we intend to do if we switch between different technologies, for example, HPLC to UPLC.
I'm really looking at that resolution aspect, which I think is causing a lot of concerns. I think the heat map that's in the 1230 chapter and the fish diagram are really good at making a start on it.
Yeah, I mean, if we think about the method operable design region, again, from a GSK perspective, we don't see much value in registering a method operable design region. We see the value more in developing the enhanced robustness, doing modeling experiments to demonstrate that where we intend to operate the analytical procedure is robust. And if you've done that, we don't see any reason why you would want to change the method conditions. Now, there might be the occasional method where one parameter is subject to variability, and you may want flexibility in that method parameter. But I would ask, maybe you've got a non-robust method. So I'm not entirely sure from a regulatory perspective why we would need an MODR, but I think the benefit is really in exploring the MODR to basically justify that the set points where you intend to operate the method is robust.
Giving people advanced understanding of where the method sits in terms of a plateau, where the risks sit on it, that optimizing the kind of pharmacopoeial control strategy for the method in the monograph, I think is a really transformational change. So one that I think Phil mentioned being excited. I'm really excited to see that, because at its heart, public quality standards have to be robust, rugged, and reliable, and that's what people need to assure quality of the products that they're testing and releasing, and ultimately quality underpins the safety and efficacy of medicines going to patients. So I think it's a really, really interesting time potentially for pharmacopoeias.